Hi, everyone. My name is Frida Teran, and I'm in the University of Iowa. In our lab, we study Dravet syndrome as our disease model, which is a devastating form of epilepsy in which children develop seizures within their first year. Most cases are caused by mutations in the SCN1A gene, which encodes a voltage-gated sodium channel. We have a mouse model with a knock-in mutation in SCN1A previously identified in 3 Dravet syndrome patients from independent families. These mice recapitulate the human disease well, in that they have spontaneous seizures and a high incidence of premature mortality due to SUDEP, as you can see in the survival curve, where death begins to occur soon after being weaned. We became interested in dietary therapies after studying the effect of a ketogenic diet on survival in Dravet mice, which is why we were approached by a colleague who studies Drosophila flies with sodium channel mutations. He discovered that fly food containing milk whey abolishes the seizure phenotype in one of his mutants. So we decided to test that in our mice. We weaned a group into a diet containing whey and another group to regular feed and monitor them with video surveillance 24/7. And effectively, we found that this resulted in a lower mortality rate from SUDEP in a dose-dependent manner. So to figure out why these mice weren't dying, we went back and reviewed videos of the 5 days post-wean, which is a period of high mortality in these mice, as you can see. And we're currently completing the analysis, but so far it seems that mice treated with milk, whey, are having less convulsive seizures. Now, how is whey doing this? Well, as opposed to ketogenic diets, whey does not cause ketosis, meaning there are likely other mechanisms involved here. Milk whey is composed of several proteins, and one of the main components is alpha-lactalbumin. And alpha-lactalbumin happens to have one of the highest contents of tryptophan. Studies have shown that diets that are high in tryptophan increase serotonin levels in the brain. Now, this is very important because serotonin has long been implicated in epilepsy and SUDEP. And studies in rodents have shown that anti-epileptic drugs elevate serotonin levels in the brain as part of their anticonvulsant action. So to check if this was the case, we fed a group of mice whey for 5 days, sacrificed them, and extracted their brain. We found that serotonin was indeed increased in the brainstem, which is where most of the cell bodies of serotonin neurons are found. So this was interesting, but it's also just a correlational finding. The next step is to remove serotonin from the equation. And that is something that we are currently doing. So we're currently depleting serotonin using multiple methods to see if that abolishes the rescue effect of whey. Another important question is whether this protective effect we see in our Dravet mice would also be seen in other epilepsy models. So we tested this in KCNA1 knockout mice, an epilepsy mouse model that is well characterized in the literature and also dies from SUDEP. And we found that it does prolong the lifespan as well, suggesting that whey could be protective in other models. But how does this translate to patients? Well, if you recall from the survival curve that showed you at the beginning, Dravet syndrome mice start dying from seizures soon after being weaned, which is also when their diet changes from mother's milk to chow. And as you can see, human breast milk happens to have a high content of whey protein. So with the help of Doug Nordley from the University of Chicago, we conducted a survey on breastfeeding patterns in Dravet syndrome patients and found that patients that were breastfed for at least 6 months or more were less likely to be hospitalized with status epilepticus. So we're hoping to publish these data soon. Another thing is that companies that make ketogenic meals and drinks are starting to come up with formulations that contain more whey protein. So our hope is that our findings may guide future clinical trials to evaluate the efficacy and safety of milk whey as a supplement for drug-resistant epilepsy. And with this, I'd like to thank the members of the Richardson Lab, especially Dr. Yu-Jung Kim, who started the experiments on ketogenic diets in our mice, as well as Dr. George Richardson, who's my advisor here, and our colleague, Dr. Toshihiro Kitamoto, who was the one that was been doing the experiments with Drosophila flies. Thank you.